So after having the knowledge of the renal corpuscle, it's necessary to know what exactly the material that is found within this renal corpuscle. So in the blood space of the renal corpuscle, that is in the glomerular capillaries, we are having blood. So this part is known to us. Here it is, what we are having is blood. But the part that is present in the urinary part, that is in the Bowman space, that part is basically a filtered product of the blood. What did I say here? Let me write in a separate space. Filtered product of the blood, and that filtered product in one word is known as glomerular, because the filtration is taking place in the glomerular. So that's why this is known as glomerular ultrafiltrate. You can call this as the first precursor of the urine. And the composition of the glomerular ultrafiltrate is almost the same like that of a blood, except, so there are a few exceptions. So how would I describe glomerular ultrafiltrate as? That this is exactly like that of blood, except, so let me write this exceptionals in a different color, except it does not have blood cells. I have probably explained what are the types of cells to be found in the blood: RBC, WBC, platelets. These are not found in this glomerular ultrafiltrate. And the other thing that is not found in the glomerular ultrafiltrate is blood proteins. So what do you mean by the blood proteins? Namely the proteins that help in the blood coagulation, fibrinogen, fibrin and all that stuff. As well as in the blood, we are having albumin protein, as well as in the globulin protein. Significantly, I would mention specifically of the albumin protein, because this albumin protein marks a threshold. Normally not to be found in the urine, but if found, that indicates a glomerulonephritic syndrome. That means a particular disease of this glomerulus. As a medical laboratory technologist, when you perform examination of the urine, you might have to detect the presence of albumin, just to confirm whether any kind of inflammation or any kind of nephritic process is going on inside this area. Okay, I must mention that somebody by the name Redmi is trying to join. So whosoever it be, I do not know. Tell that person to join in their official name. I shall definitely admit. Anyways, so these are the two exceptions. So apart from that, whatever is found in the blood are also found in the glomerular ultrafiltrate. Namely, what are they? Water, it is found in the blood. You already know. Water is found. And the dissolved materials of the blood. What does that mean? The sodium, the potassium, these are all the inorganic parts. Regarding the organic parts, the glucose, all these are getting filtered. Now, I have so far named the useful materials. So what I'm trying to say is that the materials that are of use to the body do also get filtered in this particular area. So though useful materials also ought to be found in this glomerular ultrafiltrate. But obviously, the useless material do also make its appearance in the glomerular ultrafiltrate. So what are those useless materials? The first one to name is urea. Next is uric acid. Third is creatine. Such and such and such. I mean, mostly it is urea and uric acid. So these also gets filtered. Clear? Is this part clear? The glomerular ultrafiltration. How is the filtration taking place? What are the forces behind this? That if we need to cover up, we will cover that up, but only after taking a look into the other successive stages, which finally results in the formation of the urine. Okay. So the next stage, first stage, let me put a number out here. What was the name of the first stage? The ink of this pen is very thick. Stage number 1, that is known as glomerular ultrafiltration. Stage number 2, selective reabsorption. So what does this process take place? Moving back to the diagram of the nephron again, we will find that this Bowman space leads to a tubular structure. Did you notice it? A kind of U-shaped tubular structure. Notice this. Noticed?
Yes, sir.
And this entire tube is known as renal tubule. Now this renal tubule has few parts. In a separate space, let me write down the parts of the renal tubule. Parts of renal tubule. If I draw this same diagram just to the side, I would add another detail. So this be the Bowman space. Initially, the tubules remain somewhat twisted and turned before becoming straight. And once straightened, thereafter, after a short distance, the tube gets a bit narrower. After being narrower, the tube takes this U-shaped turn and then broadens again. Continues as a straight broad part, which now becomes twisted and turned again, finally to unite with something called this straight tube. This is known as the collecting duct. And this entire thing below the Bowman space is known as the renal tubule. So putting the renal tubule and the Bowman space together, the term nephron is in use. Understood? So the nephron is divided into the Bowman space and the renal tubule, and the renal tubule is having multiple subdivisions. I'm now going to list here the different subdivisions of the renal tubule. Let me put here some numbers for the purpose of labeling. I'm labeling this part as number 1. This is number 2. This is number 3. This becomes thus number 4. This becomes 5. This becomes 6. This becomes 7. And the 8th one I have already named. So number 1, starting with the number 1, you see that it is a tubule which is highly twisted and turned. So we use the word convoluted. Convolution means this twisted, turned, and coiled appearance. This is known as convoluted. Okay. And notice that the flow of the filtrate would be in some, say, this direction. So this is an descending limb. This is the descending part. After the bend, this becomes the ascending part. This is the descending part. Understood the concept of descending and ascending?
Yes, sir.
Yes, sir.
Why have all of you kept the video off? I mean, excepting Sumana, everybody has to keep the video off. Do consider turning it off. Okay. So this number 1, which is a convoluted tubule, is thus to be termed as the descending convoluted tubule. So after this convolutions, that means this twisting and turnings and coil formations, comes this part number 2, which is a straight. So we simply replace this word descending by the word straight. Sorry, the descending word should be there because it's still having the descending character. So descending, but instead of convolution, we will write it as straight. Again, tubule. Understood? Why are we naming this? Okay. Now, this entire narrowed part, which is divided into part number 3, part number 4, and part number 5. All these 3 parts taken together are known as loop of Henle. So in the loop of Henle, we are having 3, 4, and 5 all taken together. What do we call it as? Loop of Henle or Henle, which is having the 3rd part. So the 3rd part is again having this descending character. We will call this as the number 3 is the descending character is there. So descending limb of loop of Henle. Okay. Part number 4, which actually is the turned area. Observe again in the diagram. This part number 4 is the area where the tube is taking this U-shaped bend. So this bent area, the part number 4, is the appropriate or more proper loop of what? Loop of Henle. Though 3, 4, 5 taken together can be considered as the loop, but this loop is having a loop proper or the loop of Henle and is having a descending loop, and accordingly the 5th one to be termed as the ascending limb of loop of Henle. Understood the concept of loop of Henle? It's very difficult. Understood. Moving from the 5th upwards, you notice we are having this area called 6, which is a straight part. Noticed? The 6th area is again a straight, similar to the area number 2. But this time, the course of the flow of the contents inside the tubule will be in the upward direction. That means this is having this ascending feature. Correct. So the 6th one is thus to be termed as ascending. So what was the name for the part number 2? That is descending straight tubule. Similar to that, since the part number 6 is present, we'll also call this as a straight, but this will be a ascending straight tubule. Clear? I'll be writing the name out here. Descending. Is it descending? No. It is what? Ascending. Part 6 is ascending, moving upwards, right, Sumana? So this
Yes,
is
sir.
ascending. Ascending
Yes, sir.
straight tubule. Now, as the part number 6 ascends more upwards, notice that in the part number 7, we are having this tubule again multiply twisted, turned, and coiled. So the part number 7 would thus be having a name. And the name for that part number 7 will be obviously convoluted, right? Similar to the part number 1, we'll be having this convoluted, but this convoluted is of the ascending sides. So we call this as the ascending convoluted tubule. So see all the paths that were drawn. There were a total of 7 paths out here. And the name of those paths are written out here. So the first name is out here, descending convoluted tubule. And the last name, which also is a convoluted, is out here. That is the ascending convoluted tubule. Clear? So the process of urine formation continues along the entire length of this tubule. Difference is that along the parts say 2, 3, and 4, or more appropriately 2 and 3, that is the descending part, we have the second process or the second step in the process of the urine formation, and that process is selective reabsorption. Now let me tell you in short that what happens in reabsorption. Just now we have noted that during ultrafiltration, all the compositions of the blood enters into the ultrafilter. This is what is written here, right? Ultrafilter is having all the compositions of the blood except the blood cells, the blood proteins. Okay, so these are the 2 exceptions. That means the filtrate is having all the useful and necessary materials, namely the glucose, the water, the sodium, the potassium, all the stuffs. And these we are not supposed to eliminate because these are useful materials. That means they needs to be returned back to the body or to the blood. Understanding? So as this glomerular filtrate, I would write here as GF, standing here for the glomerular filtrate, is flowing downwards through the tubules. I have said that entwined around the tubules are even the blood capillaries. That is these are the peritubular capillaries. So as the filtrate is moving along this tubule, there is an outward movement of materials. So from the urinary space to this blood, there is a movement of what? Movement of all the useful materials. Useful materials is moving out into this peritubular capillaries. So that means what we can say that this is a kind of reabsorption, absorption back. So that means reabsorption. But this reabsorption is not universal. That means this reabsorption is highly selective. That means we are not supposed to reabsorb the waste material. What are those waste material? That is written here. This urea, uric acid, and the creatinine. This shouldn't get reabsorbed. But what should get reabsorbed is the useful material. That is the sodium, potassium, glucose, potassium, all this stuff. Water. This should get reabsorbed. So this entire process of reabsorption goes on in this area. Clear? Is the concept clear?
Yes, sir.
Yes.
Coming now to the last stage, the third stage, which is known as, I have not yet written that name of the stage on the board. Here is where I should write. The third stage is tubular tubular secretion. Now, what is tubular secretion? See, while the filtration was going on in this particular area, while the filtration was going on, it is not possible actually to eliminate all the waste in this area. Elimination is possible through filtration. So this area doesn't ensure 100% elimination of all the metabolic waste. That means the blood that is entering into this efferent artery out here and is making another capillary, that means this peritubular capillaries out here, this peritubular capillary, that blood has still some residual amount of waste. Did you get this point? The blood that is entering into the glomerulus is not getting filtered of its waste 100%. Some residue is being carried away by the efferent artery, quite naturally, because the process is very fast. Being that very fast, not always it is necessary that all the waste materials would move into the Bowman space. Some part of the waste might remain inside the blood of the artery as some residue. Are you understanding? But that residue needs also to be eliminated. And that elimination process occurs in this area, where the movement now takes place from this blood space into the urinary space. Remember the direction here where the reabsorption was going on. So here it is the area for the reabsorption. The movement of the material was from away or out of the renal tubules into the blood. But here, where this tubular secretion, let me narrow down the ink. Tubular secretion is going on. In the process of the tubular secretion, materials are entering into the tubular space from the blood. Which materials are entering? The residual waste. But in the tubular reabsorption, what is happening? What is being moved out of the tubules into the blood? These are all useful materials. Useful materials. What are they? To name few such useful materials. It should be, say, water, glucose, sodium, potassium. I'm just writing the formula, okay, because of lack of space. Sodium is having the formula Na, that is natrium. I think you have learned it by now. These are the basics of the chemistry. Biochemistry is already introduced to you. So these useful materials get reabsorbed back, and the residual waste are sent back to the urinary space. So now at this junction, what is this junction named as? The junction between the nephron and the collecting duct. I'm speaking of this junction. So this is where the nephron is ending, and the nephron is joined here with the collecting duct. So whatever is collected in the collecting ducts, all those materials is the finally formed urine. Understood? So whatever is collected here is the urine. Okay. So this briefly describes the physiological aspects of the urine formation. Very briefly though. Now, there obviously is a confusion that from the diagram of the kidney, I have taken you directly to the nephron. The next question thus arises is, how are these individual nephrons organized or remain organized in the single kidneys? So to answer that question, we will take one kidney and make a section of that kidney, a kind of coronal section. I think you know what is coronal section? Something like this. So if we make a coronal section, what you will see inside the kidneys, so let me draw the kidney again. What you will see inside the kidneys is that there are multiple line kind of appearance. And to the periphery of this line kind of appearance, there are multiple dots. Some of this dotted area even projects in between these lined areas. These are the common features that we will note as we make a section of the kidney. Now let me name these areas one by one. This outside area, which is having all this dotted appearance, this is known as the renal cortex. The inside area, which is having these multiple lines, this is known as the renal medulla. So these are the two principal broad divisions of the structure of the kidney, cortex and medulla. I have still not discussed where is the nephron present in the structure. Now, if we enlarge this view, let me now show you how it is organized because not much time is left. If we enlarge this view, so these dots will become big. So at one point of time, these dots will appear as having, I'm erasing one dot and trying to show you that how will be the exact appearance, as having a shape of the Bowman's caps, inside which there is this tuft of capillaries. What I say is that, see, as I lower it down, this appears very small. And as it appears very small, at one point, even now, you can see this as a dot. Notice my cursor. Doesn't it appear like a dot? Yes or no? Please do confirm. Yes.
Yes, sir.
Now, on increasing the magnification, so if I use a microscope to see the structure, then under the microscope, I will see it on a higher magnification that the structure is having the similar to the shape of the Bowman's capsule and the enclosed glomerulus. So these two structures taken together is known as what? Is known as the renal corpuscle. So these dots, all these dots out here are basically renal corpuscles. Clear? So renal corpuscles are one part of the nephron, which itself is having both the urinary part as well as the blood part. Now, what are these lines? If I draw the entire nephron back from the Bowman space, what would be the diagram? Are you getting the concept? Does it now look like a nephron, which is having the entire renal tubules also? Is this stretch not the renal tubule? That was the renal tubule that I showed you in the diagram just beside it. Forgot. I'm speaking of this particular diagram where I have shown the renal tubule. So since it is tubular shape, when it is made very small, would it not look like small lines? If this appears as a pen, if I take it farther, at one point, would you not find this a line? Because the more farther it moves, the smaller it becomes. And at one point of smallness, this entire tube would appear just a single line. You do understand the concept? Just like, where was the diagram? This one. See, there was a single line. Can you see this line? Even in this level of magnification, the line is seen. Notice this line out here. Single straight line. Observe my cursor. Now, if I enlarge it even more, see it at multiple such enlargement, you find that this line appears now like a tube. So whatever the lines that we see in this middle part, all these lines, which color is it of? All these lines are basically the renal tubules. So this renal tubules and the renal corpuscles taken together is known as what? It's known as, or the structural and functional unit of the kidney, that is kidney nephrons, or simply nephrons. As I said, that in few areas, in between this projections, this macroscopic appearance, this entire appearance is known as renal pyramids, because it looks like a pyramids, simply because of that. But this renal pyramids themselves are made up of multiple lines, and each lines is the contribution of either the renal tubules, or in some areas it can also be of the collecting duct. So this was the collecting duct. I mean, that same image of the nephron, when placed in the gross macroscopic structure of the kidney, this is how it would look. So this is how multiple nephrons are organized inside the kidney. Understood? So I have given you, let me now summarize the entire session, this 2-class session. Okay, the video upload in the YouTube is almost over. It can be published, but I'm going to publish after this session is over, because only 1 minute 15 seconds left. Let me now summarize what I have taught you today. I've taught you the entire excretory system. I've discussed the physiology of the urine formation, the organs of the excretory system. Apart from the urine, whatever other excretory waste we do have, that also I have discussed. I've discussed the urinary system being the most considerable part of the excretory system. Now, the urinary system's organs are already discussed. The macroscopic structure of the kidney is presently on the board. The microscopic structure of the kidney is just like the nephron. Okay, so here the nephron is shown in detail. And this nephron of the kidneys are basically the urinary part. But the urine is the product of blood after getting filtration. So this is how the entire filtration and the other 2 steps, selective reabsorption and tubular secretion, is shown in the diagram. So with that, for the last 1 minute, the entire session comes to an end. It will be recorded and uploaded soon in the YouTube. Do follow there. This is the entire boardwalk of today's class. Either you can take a screenshot, or you can follow my YouTube.